Daniele and I present the work Development and Characterization of a CD4-GFP chimeric protein. Defining some concepts. First, biopharmaceutical are a class of therapeutic drugs that are manufactured through biotechnology process, such as recombinant proteins produced by genetically modified cells using fraction vectors generated with recombinant DNA technology. A promising biopharmaceutical are fusion or chimeric proteins, which are formed through the genetic fusion of two or more distinct entities, to improve the pharmacological characteristics and directing the protein action. Thus, a typical chimera is formed by a dominant effector and an auxiliary or ligand, which directs the action of the dominant effector. The chimeric protein can be used in the study of development of products for the managed AIDS, a pathology caused by HIV, which affects cells with the CD4 receptor in your surface. Thus, the work aims was constructing and characterizing a chimeric protein harboring the soluble human CD4 domain bound to the GFP reporter to evaluate the potential for the use in HIV infecting cells diagnosis. The general methodology consists in cloning the CD4 GFP as expressing one fastback vector. With the fastback CD4-GFP, we transform the E. coli DH10 back, which contains the wild-type back medium, to make a recombination generating the CD4-GFP back medium. With the back medium CD4-GFP, we transfer the insect cells SF9, generating a baculovirus, which is used to produce the recombinant proteins that contain histidine tag in your composition, so is purified by IMAC chromatography. With the purified protein, we made the characterization and functional assay. To characterize the CD4-GFP, we first analyzed our purification, performing a CDS page densitometry and protein quantification. Thus, we see a protein with the molecular weight corresponding to expected, with the 18% for relative purity. We also observed that our purification global process recovery was 6.3%. Performing a dot-blot assay with the fractions from the protein purification with the antibodies directing to CD4 to confirm the presence of this domain. We then observed the presence of labeling in all fractions being considerably stronger in the purified fraction. So we concluded that the protein seen in the gel was indeed our target. If your protein had both functional domains, we performed an immunocapture analysis with polystyrene bits associated to CD4 antibodies. We used as control a protein GFP. Analysis was by flow cytometry, where we could observe a significant increase in the intensity of GFP on our immunocapture bits included with the CD4 GFP, thus showing the functionality of both chimera domains. We made two main analyses regarding the amount of protein and the time needed to observe this marking. In this assay, it was possible to see if using 5 micrograms for the last 30 minutes, we can select cells expressing the viral receptor. Therefore, the CD4-GFP chimeric protein has a potential for use as a tool for the development of diagnostic assays and research of new HIV antiviral drugs that act in a viral interaction with the target cells. I'd like to say thanks to FAPESP, CNPq, and CAPES for supporting this work, and Ellen Bill and CNPN for the use of their facilities.